Hey everyone, this is Abdulrahman Masood, and today I'm going to be explaining the first physiology lecture of the endocrine system. So first of all, why do we study the endocrine system? Why is this endocrine system very important to us? We already have 11 systems in our body, so why do we need this extra system? What's the function of this body, system? The main function of this system is to regulate everything inside our body, to regulate all the functions, all the relationships, to control everything. So basically, maintain the homeostasis in our body. You may think we already have a system for that. We already studied the nervous system and it literally had this exact same function. But the difference between the actions that the nervous system control and the actions that an endocrine system control is the speed at which I want these actions to happen. The nervous system is very fast. But the endocrine system is very slow and steady. We are going to talk about the differences between the nervous system and the endocrine system throughout the lecture. But right now, I want to focus on a new term to us. It's called endocrinology. Now, what is endocrinology? In the endocrine system, we have these glands, and these glands secrete this hormone. And this hormone will go, this chemical substance, this hormone is going to go all the way in the bloodstream until it finds its target cell, where it's going to have the receptor. The receptor is going to have this target cell, and then this hormone is going to bind to this target cell, and it's going to form an action. And this whole process is what we like to call the endocrinology. So endocrinology is the study of hormone receptor interaction and signaling pathway due to this binding. So how did they discover this system? What's the history behind the discovery of this incredible system? There's a scientist. His name is Arnold Berthold. I'm not really sure if I'm pronouncing this correctly. But anyway, he did an experiment. He took a male chick, a rooster, and then he castrated this male chick. So he removed the testicles, and then he started examining the differences in the behavior and the characteristics of the male rooster, and everything changed. This rooster became less aggressive. The comb decreased, which is a male characteristic, and he became less. His sexual desires decreased. And what was crazy to them is that when they re-injected this rooster with the testicular fluid, everything came back to normal. He started becoming more aggressive, the comb grew, back, and his sexual desires came. So what they have concluded is that, this testicle, it releases a certain substance, which they didn't know what the substance was, and this substance, affects the body, whether it's characteristic or behavior, it affects the body. And later, in 1903, Starling and Bayliss found out about the hormone secretin, and they were the first to use the term hormone. So yeah, now I'm just going to read everything. History. Early experiment by Arnold Berthold on roosters. Berthold found that rooster's comb is an androgen-dependent structure. Following castration, the comb atrophies, it decreased in size. Aggressive male behavior disappears, interest in hens is lost. Hens are the female chicks. Also, he found that these castration-induced changes could be reversed by administration of crude testicular extract, by pure testicular extract, or prevented by transplantation of the testes. Starling and Bayliss demonstrated that secretin stimulates pancreatic secretions. They are the first to use the term hormone in 1903. I've been saying from the beginning of the lecture is that the whole function of this endocrine system is for all the systems and the organs to communicate with each other. But what are they using to communicate with each other? I use the internet to communicate with my friends. So how are these organs are communicating with each other? The first way that they're going to communicate with each other is called autocrine. Auto comes from the word self, which means that, a cell releases a signal and then it affects its own self. There's a receptor on the cell, so it's affecting itself. Para comes from the word alongside. So it's the same idea, but the receptor is found, alongside the cell of release. So the effect is happening on, the nearby tissue. The neurotransmitters is something that we have studied before. They're released from neurons, and then they go to the presynaptic membrane, postsynaptic membrane, and then another neuron, and then another neuron, and then it is released to activate receptor on an adjacent cell. Then the endocrine, the hormones, these are, what we're focused in the system. As we said, they're released from endocrine cells into the bloodstream, and then they react on a very, very, very distant receptor. And then there's a term that I want to talk about. So the neuroendocrine cells, these are just neural cells that have been modified to produce and release release hormones. So these are found in 3 different parts in our body. First of all, they're found in the hypothalamus, they're found in the posterior pituitary glands, and they're found in the medulla of the suprarenal gland. There are certain cells that are called chromaffin. And these cells are basically neuroendocrinal cells. They produce and they release the the hormone. So chemical messengers, the activities of cells, tissues, and organs are coordinated by chemical messengers. Autocrine substances affect the same cell of origin. Paracrine work locally, affect the neighboring cells. Neurotransmitters released from neuronal terminals into a synaptic gap to activate receptors in adjacent cells locally. Endocrine hormones, chemicals released from glands into bloodstream to act on distant receptors. Neuroendocrine, neurohormones, hormones released from neurons and transported via bloodstream, mainly found in the hypothalamus, posterior pituitary, and chromaffin cells in the medulla of adrenal glands. All right, and now for the differences between the nervous system and the endocrine system. So as we said before, the nervous system is very fast. And why is it fast? Because it's anatomically all connected. So we have, a neural cell, and then a synapse, and then another neural cell, and then another neural cell, and then it keeps going until I find, my site of action. And but in the endocrine system, it's a much slower control. And the receptor distribution is what identifies where I want this action to be. So let's say I want the action to be over here, so I'm going to have a receptor over here, but I want an action to happen over there, so I'm going to have a receptor over there. And the cell that has the receptor is what we call the target cell. It's the cell that it's basically the target cell. And how these receptor and hormones attaching, it's just the good old key and chain action. It has to do with the shape. The shape of the receptor has to fit exactly in the shape of the hormone. So nervous versus endocrine. Nervous system, fast control due to anatomical connections between neurons, synapse, which determines specificity. Endocrine system, slow control, receptor distribution determines specificity. Only cells with hormone receptor will respond. Target cells. Target cell that express a specific receptor for the hormones. It can express receptor for more than one hormone. Yeah, I forgot to say that point. The same cell could have different hormones acting on it. You just gotta have the receptor and you're good to go. Accommodation in shape, conformation, shape of the hormone is suitable to its receptor. Classes of hormones. So we have peptides, we have steroids, and we have modified amino acids. So basically the whole idea over here is, you have to understand which ones are hydrophilic, which ones are hydrophobic. So for the peptides, peptides are proteins, so they're very hydrophilic. They're water loving, but they're also lipophobic. They're lipid hating. So they cannot cross the lipid bilayer. So there has to be a receptor for the peptide to enter the cell. And then there has to be a second messenger in order to, induce the action. But with the steroids, these are they're lipophilic. They're hydrophobic, so they're water hating, but they love the lipids, so they can cross the lipid bilayer of the cell, and then so they don't need a receptor on the lipid bilayer, so their receptors are usually found inside the cell or even inside the nucleus. Yeah, that's pretty much the basic idea. All right, so peptides. So what are peptides? Basically they're just a bunch of amino acids that formed a protein. And we've already talked about the synthesis of protein before. It's very simple actually. There's a transcription process, and then there's a translation process in the ribosomes. And then this translation process produces this precursor inactive form of peptides. They're called pre-pro hormone, and then this pre-pro hormone has to go to the endoplasmic reticulum in order to be a little bit modified to give us the prohormone, and then this prohormone further goes to the Golgi apparatus to be further modified to give us the active form, and then obviously stored in a vesicle. And then upon stimulation, whether the stimulation was calcium or cyclic AMP, this hormone will be released, this peptide hormone will be released into the bloodstream. And since it's hydrophilic, it doesn't need a carrier protein. Then it's going to have a short half-life. If you don't know what a half-life is, it's basically the time that it takes for the hormone to lose half of its physiological activity. So yeah, since it doesn't have the carrier protein, so its half-life will be decreased. This is going to be a little bit different in the steroids. So peptides, they are made of chains of amino acids. They're hydrophilic, water soluble, can be small, 3 amino acids as thyrotropin releasing hormones, or large, 100 amino acids. Now others, adrenocorticotropic hormone, the ACTH, growth hormone, prolactin, gonadotropin, insulin, glucagon, and parathyroid hormone. Now the receptor, that's the good part, in the plasma membrane, it can't cross it, needs second messenger inside the cell. Synthesis starts in the nucleus, transcription, translation in ribosomes to produce proteins in inactive, which is inactive, precursor protein, pre-pro hormone. Pre-pro hormone has a signaling sequence, pre-sequence, directs it into the endoplasmic reticulum, undergoes activating step by cleavage of pre-sequence. Remaining prohormone will be targeted towards the Golgi apparatus. Pro-sequence is cleaved, stored in vesicles. Upon stimulation, the vesicles release the hormone from the cell of origin via exocytosis. Exocytosis is just basically when the vesicles fuse with the lipid bilayer, and then it's going out into the bloodstream. Blood transportation, no need for carrier proteins, short half-life, life cleared faster. Hormonal half-life, the time it takes for the hormone to lose half of its physiological activity. Now, as for the synthesis and the blood transportation of steroids, these are lipophilic, so they can just directly diffuse out of the lipid bilayer, and they're not stored in the cells. But since they are hydrophobic, so they have to be bounded to this carrier protein. So this carrier protein will give it a longer half-life and will decrease the metabolic clearance. So it's going to stay for a really long time in our bodies. And there is a uniform distribution. Why there is a uniform distribution? Because it's found everywhere in the bloodstream. Yeah, so modified lipids. So what are steroids? They are basically modified lipids made of cholesterol, lipophilic. Yeah, steroids are modified lipids made of cholesterol. They're lipophilic, they're water soluble. Includes adrenal hormones, the cortisol, the aldosterone, and the gonadal hormones, the estrogen, progesterone, and testosterone. Receptors, the location of the receptors is inside the cell, can cross the plasma membrane, affects the genetics of the cell, transcription and translation, since the receptor could be inside the nucleus. No need for a second messenger. Synthesis and blood transportation, not stored inside the cell, released directly by diffusion in bloodstream bound to carrier proteins, long half-life, decreased metabolic clearance, good reservoir for hormones, and uniform distribution. Yeah. Right, and now for modified amino acids. Modified amino acids are just modified tyrosine. Tyrosine is an amino acid. And this category is very simple. Some of them are closer to characteristic to the peptides, and the other are closer in characteristics to the steroids. We have the catecholamines, which are closer, which are similar to the peptide hormones, and then we have the thyroxine, which are similar to the steroid hormones. So yeah, the catecholamines, they're similar to the peptide hormones, so their receptors have to be where? Yes, their receptors have to be in the plasma membrane, and they should have their half-lives should be very short. But with the thyroxine, since they are similar to the steroid hormone, so their receptors have to be inside the cell, specifically in the nucleus, and they have longer half-lives. It's that simple. So modified amino acids, mainly modified tyrosine, include catecholamines from adrenal medulla, thyroxine from the thyroid gland. Receptors, catecholamines are similar to peptide hormones in plasma membrane, thyroxine as steroid hormone inside the cell, specifically in the nucleus. Blood transportation, catecholamines are similar to peptide hormones, half-life few seconds, minutes. Thyroxine as steroid hormones, half-life months. Right, so to keep the hormone at normal levels in our body, we have to do a balance between the secretion and the excretion. So if we secrete a lot of this hormone, we're just going to end up having a lot of this hormone, and obviously this is an imbalance. But before we talk about the mechanisms for controlling the hormone secretion, we have to know, how is this hormone secreted? What way is it secreted? Is it secreted in a continuous manner or in a pulsatile manner? And the answer to this question, you guessed it, it is in a pulsatile manner. So what does that mean? We don't get surges of hormones being released. So let's imagine that I am the gland and I'm trying to secrete this hormone. I'm not gonna, secrete all of it at once, because if a gland secretes this hormone at a steady manner, but like in surges, in a huge amount, in a continuous manner, all of these receptors are going to form this sort of resistance. So their effect is going to decrease. So what we do is that they secrete it in a pulsatile manner. So it goes up and then it goes down. It goes up and then it goes down. And yeah, that's... So yeah, pulsatile hormone secretion. Most of hormones are secreted in pulsatile way, which help in full effect of hormone. Episodic rhythmic way of secretion, burst-like, rise in concentration at time period and then go down at another. Repetitively, this cycle continues, peak and bottom. Example, secretion of corticotropin releasing hormone. And as we said, anything that says releasing hormone, then it's obviously from the hypothalamus. So example, secretion of corticotropin releasing hormone from hypothalamus within 10 minutes. reaches its maximum level, then drop reaching its minimum level. So 10 minutes of up, and then it goes down. If it stayed further after the 10 minutes, and if it stayed continuous after the 10 minutes, the effect is going to be down. The receptor is going to form the resistance, and the effect is just going to decrease. So it has to go down and then goes up. Another example, effect of gonadotropin releasing hormone, you guessed it, from the hypothalamus on the pituitary LH and FSH. Pulsatile way, normal levels of LH and FSH, if continuous and steady infusion of gonadotropin releasing hormone reduced or lost levels of LH and FSH. There are many methods of controlling this hormone secretion, and the first thing that we're going to talk about is the negative feedback mechanism. Now, that's a mechanism that we've talked about a million times before, but I'm going to give you an example from the endocrine system, and you're going to understand it very easily. So we have the hypothalamus. So the hypothalamus is the big boss, as Ayash said. The hypothalamus releases this hormone. It's called thyrotropin releasing hormone. So this thyrotropin releasing hormone goes to the pituitary gland, the boss, not the big boss, the boss. And then this the pituitary gland will release a thyroid stimulating hormone. Now this thyroid stimulating hormone will go all the way to the thyroid gland, and then the thyroid gland will produce its hormones, the T3 and the T4. Now let's say that my body have produced a huge amount of T3 and T4. I can't keep on producing this, because then I'll lose my balance between the hormones. So what happens is that these T3 and these T4 will go all the way back to the hypothalamus and the pituitary gland and tell them, hey, stop, chill. Just don't release your hormones. And this is what we call the negative feedback. So negative feedback mechanisms prevent the overactivity of hormones. Most common help in homeostasis. Example, hypothalamic pituitary thyroid axis. Hypothalamus secretes the TRH, which is the thyrotropin releasing hormone. The TRH stimulates pituitary gland to release the TSH. The TSH stimulates thyroid gland to release T3 and T4. If T3 and T4 levels increase in the blood, prevent further release of thyroid hormones by inhibiting the secretion of TSH and TRH. But in the opposite feedback, it's literally everything but reverse. It's the opposite of homeostasis. We're trying to reverse everything. The release of, the hormones are going back to the hypothalamus and the pituitary gland and telling them, oh, I want more of this. So I'm going to give you, again, another example from the endocrine system. So the hypothalamus releases this hormone. It's called gonadotropin releasing hormone, and this gonadotropin releases hormone goes to the pituitary gland and lets us secrete the LH, the luteinizing hormone. This LH will go to the ovaries and let the and lets the ovaries produce estrogen. Now, negative feedback happens throughout the whole menstrual cycle. So if the estrogen increases, is going to go back to the hypothalamus and the pituitary gland and tell them to stop, except for the 2 days before ovulation. Just 2 days before ovulation, the production of estrogen will go back to the hypothalamus and the pituitary gland and tells them, hey, I want more. So this is where the luteinizing hormone surge comes in for the ovulation to happen. So there is technically a reason for this positive feedback, and it's just for the ovulation to happen. That's all. So yeah, positive feedback control, produce surges of hormones, amplify hormone secretion. Example, hypothalamic pituitary ovarian axis, HPO axis in female. Hypothalamus secretes gonadotropin releasing hormone. Gonadotropin releasing hormone stimulates the anterior pituitary gland to release the LH. Stimulates ovary to secrete estrogen. During the menstrual cycle, estrogen exerts a negative feedback on pituitary gland or even gonadotropin releasing hormone from the hypothalamus, except 2 days before ovulation. Estrogen exerts a positive feedback action on pituitary gland, more release of LH needed for ovulation. So not only the nervous system and the endocrine system are very similar, but actually sometimes there is a little bit of a cooperation between them. They work together to, control our, homeostasis. Sometimes when there's a sudden need for hormone release, that's the key word, sudden. That's why we're needing the endocrine system, the nervous system, because the endocrine system isn't used to sudden actions. It's used to, more slow action, but the endocrine system is a master at sudden actions. So what happens here is that, when a mother is trying to breastfeed her son, and then as soon as the touch receptors on her breasts feel the suckling of her child, this stimulates an action potential, and this action potential, is transmitted through neurons all the way to the hypothalamus, and then it tells the hypothalamus to produce the oxytocin and the prolactin. If we're gonna wait for the endocrine system to do that, it's gonna take a lot of time, but the nervous system is really fast. So yeah, that's pretty much it. Neuroendocrine reflexes, cooperation between nervous system and endocrine system when there's a need to sudden hormone release. Example, secretion of oxytocin during breastfeeding. Suckling process stimulates the pressure, sensitive receptors in the breast mammary glands of feeding mother. Action potential, rapidly transported towards the hypothalamus, rapidly stimulate pituitary gland to release oxytocin and the prolactin. And just to clarify everything, oxytocin is actually produced in the hypothalamus, and it is stored in the posterior pituitary gland, and the prolactin is produced by the anterior pituitary gland, and the oxytocin and the prolactin are both needed for lactation. Now, as for cyclical variations in hormone release, hormones are released at different times of your lifetime, whether it's during the springtime or the daytime or at night. But your release of hormone is totally affected by the time of the day, the time of the month, the time of the year. So hormones increase at a specific time, decrease at another. Seasonal changes, estradiol, which is a female, which is practically estrogen, release varies from season to season, so increase during late spring and decrease in winter. And this causes many problems in relationship problems, because the estradiol increases during the springtime and decreases during the wintertime. So their sexual desires will change throughout the season. The various stages of development and aging, growth hormone reaches its highest levels during puberty and then keeps declining with age. Yeah, so, the responsibility of the growth hormone is to make me grow. So during puberty, I need a lot of growth hormone because I want to grow. But then as I get older, I'm not going to grow any taller. I'm not going to grow. So I don't need this growth hormone. So as I'm aging, the growth hormone will decrease. Diurnal, daily cycle, and sleep, circadian rhythm. Cortisol shows high levels during early morning and minimum levels during late evening in a normal person who wakes up in the day and sleeps at night. Changes by day and night. So yeah, if you sleep early and you wake up early, so at day and night, in the day, you'll have high levels of cortisol, and in the at night, you're gonna have very low levels. So right now I have super duper low levels of cortisol because it really is very late at night. So it changes. So changes by day-night shift. So if you flip your sleeping schedule and you sleep during the day and you wake up during the night, the levels will change. So technically what determines your cortisol levels is your sleep schedule. So yeah, basically it increases as soon as you wake up and then decreases before you sleep. that's what it is. Growth hormone reaches its maximum level during the first 2 hours of sleep, and that's exactly why bodybuilders like to sleep, because they're trying to rebuild the muscles that they have destroyed in the gym. So they're trying to sleep so that their body will produce this growth hormone, so that they can rebuild everything that was destroyed in the gym, so that they can grow bigger and more strong. Not only our bodies control the amount of hormone that is found in our body, we can also change the density of the receptors according to the intensity of the response that we want. So let's say there's a huge, there's a very, 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 very weak response in our body. Maybe there is enough amount of hormone, but there isn't enough amount of receptors. So this whole binding situation isn't really happening. So what we do is that we do receptor upregulation. We increase the density of these receptors. So right now we have increased the possibility of these hormones binding to these receptors and hence causing an response and an action. But in receptor downregulation, it's the complete opposite. There is a strong response, but I want to decrease that response a bit. So what I do, I just do receptor downregulation. I decrease the density of these receptors, so the possibility of the hormone binding to this receptor have completely decreased. So interaction between the hormone and its receptors, we have two mechanisms: receptor upregulation and receptor downregulation. In receptor upregulation, weak response to the hormone can be enhanced in the target cell by increasing the number of receptors, stronger response. Receptor downregulation, desensitization is another way of saying it. High receptor density and strong response can be reduced by decreasing the number of receptors. Decrease or diminish response back to normal. Not only these hormones are going to interact with the receptors, but they're also going to interact with each other. And there are three main mechanisms in which they interact with each other. The first one is permissiveness, synergism, and antagonism. Starting with I'm just going to give you a little idea about each and every single one of them, and then I'm going to talk about them in detail. So permissiveness, and as its name suggests, I needed permission in order to do my job. So let's say I'm a hormone, and you're a hormone, but I can't do my job unless I take the permission from you. So yeah, that's kind of the main idea. In synergism, let's say again, I'm a hormone and then you're a hormone. My effect is 1 and your effect is 1. But when we work together, 1 plus 1 does not equal 2, it equals 3, 4, or 5. So our effects combined are more than my effect individually and your effect individually. With antagonism, one is opposing the other. Each one is trying to do something very different. I'm trying to increase something, but you're trying to decrease something. So yeah, we're trying to antagonize each other. Going back to permissiveness, one hormone needs another to exert its normal activity. Epinephrine alone, weak lipolytic effect on adipocytes, so release of low quantity of free fatty acids into the bloodstream. The thyroid hormone, it has a weak effect as well. But together, the thyroid hormone increases the beta-adrenergic receptors, so the epinephrine enhances the amount of released fatty acids into the blood. All right, so the idea over here is that the thyroid hormones will increase the beta-adrenergic receptors. Now, what are these receptors are responsible for? They're responsible to let the epinephrine enter. The epinephrine cannot enter with these beta-adrenergic receptors. So the presence of the thyroid hormone it gave permission for the epinephrine to enter the cells and then enhance the amount of fatty acids released into the blood. That's it. Synergism, more than one hormone producing the same effect on a target cell, but the combined effect is greater than the addition. No need for other to work. Yes, they're just trying to make it clear that this is not permissiveness. This is synergism. Cortisol, glucagon, and epinephrine are all hyperglycemic, so they're trying to increase the glycine, the sugar level in the body. But each one has a weak effect. Altogether, the effect will be much greater. So yeah, let's say the cortisol level, the action of it is over here, and then the glucagon is over here, and then the epinephrine is also over here, but their addition is not this, it's actually this. It's their addition is much greater than each effect individually. Again, 1 + 1 does not equal 2, it equals 4, 5, 6, 7. Antagonism, two hormones work against, opposite to each other. Insulin decreases the blood glucose while the glucagon increases it. So it's currently 4:22 in the morning, and honestly, I'm really tired to continue the lecture. There's just one page left, and inshallah, I'll do it tomorrow. I don't know. I hope this is beneficial. All right, and now for the mechanisms of hormone actions. We've already talked about the different types of hormones that are found in our body. We said that there are hydrophilic hormones and hydrophobic hormones. Their mechanism of actions obviously is going to be different, and it all going to depend on whether this hormone can cross the lipid bilayer or it can't. So starting with the hydrophilic hormones are also lipophobic, which means that they cannot enter the lipid bilayer unless there's a transmembrane receptor. And then it needs a second messenger. But if you're lipophilic, then you're not going to need a receptor that is found in the cell membrane because you already can cross the lipid bilayer. But right now, with the hydrophilic hormones, I want you guys to look at the photo, and I'm going to explain everything that is found in the photo, but keep on, focus on the photo and try to soak everything in. So, first of all, the hormone is going to bind to the receptor. And this receptor, the G protein coupled receptor, is found on the transmembrane. Now, why is it called G protein coupled receptor? Because it is bound to this protein, this GS protein, that is made up of 3 units: the alpha unit, the beta unit, and the gamma unit. And the alpha unit has a compound, the GDP. As soon as the hormone binds to the receptor, the G protein coupled receptor, this GDP that is found on the alpha unit is going to change into GTP and is going to detach from the receptor. Now it's going to detach from the receptor and it's going to bind to another receptor, the adenylate cyclase. This binding is going to change the ATP from the CAMP, the cyclic AMP. So right now we have our second messenger that we've been looking for all along. Now the cyclic AMP is going to activate the protein kinase A. Now protein kinases, what do? They phosphorylate other proteins to activate them. So all we're trying to do is to find the cyclic AMP so that the cyclic AMP can activate the protein kinase, and then this protein kinase is going to it's going to phosphorylate the proteins and activate and induce the action. And yeah, that's pretty much it. I'm just going to read it through so that you'll understand even more. So adenylyl cyclase AMP system: hormone binds to receptor, activation of G protein cytosolic site of membrane. It's called G protein coupled receptor. G protein, the trimeric compound of 3 subunits, alpha, beta, and gamma. In active state, binds to GDP and upon activation. Alpha S subunit detaches from the complex and GTP is attached. And be careful, it is alpha S, the alpha stimulatory. Alpha subunit activates adenylyl cyclase, which works on ATP, converting it to cyclic AMP, the second messenger. Cyclic AMP activates protein kinase A, protein phosphorylation, and hence the response. All right, and now for the second mechanism. So the second mechanism is very similar to the first. It's almost the same thing. All we're trying to do is to let this hydrophilic hormone bind to a receptor that is found on the membrane and then produce a second messenger. I want you guys to look at the photo very carefully, and I want you to soak in all the information. So first of all, the hormone is going to bind into a protein, a G protein coupled receptor. And this G protein coupled receptor has a G protein attached to it, which consists of alpha, beta, gamma subunit. And the alpha subunit has a GDP, very similar, right? But this one is a different one. Upon the activation, upon the binding of the hormone to the receptor, the alpha subunit is now going to detach, and the GDP is going to be changed into GTP. But right now, this is not alpha S, this is alpha Q, because as I said, this is a different type of protein. So now the alpha Q is going to go all the way to a different receptor, or a different enzyme actually, the phospholipase C. Now this phospholipase C, and as its name suggests, it's a phospholipase, so it's supposed to work on a phospholipid. And it's going to work on a phospholipid called PIP2. This PIP2 is going to produce the DAG and the IP3. Now, the DAG and the IP3, these are our second messengers. The DAG, the diacylglycerol, which is just a fatty acyl chain attached to the central glycerol backbone, and the IP3, the inositol triphosphate. This is what we're looking for. And these two second messengers, these are going to induce the actions that we want, but they're going to do them very differently. So the IP3 is going to bind to its receptor on the endoplasmic reticulum. And as we all know, the endoplasmic reticulum is full of calcium. So there will be calcium release, and this calcium release is going to induce the response that we want. But for the DAG, this will activate the protein kinase C, not the protein kinase A. Now, the protein kinase C, it's going to do phosphorylation and hence the response. Now, you may ask, why do we have, two different mechanisms? At the end of the day, they're all hydrophilic hormones. But sadly, there's a list, and this list we have to memorize. Each hormone uses a different mechanism of action, and we have to memorize which hormones use the first mechanism and which hormones use the second mechanism. I'm going to insert them at the end of the video. You can just screenshot them and memorize them. Right now I'm just going to read everything. Phospholipase, CIP3, calcium plus 2 system. Hormone binds to receptor activating G protein, different type. Alpha subunit detaches from the whole complex, and GTP will bind to it instead of GDP. Alpha Q subunit activates phospholipase C, the enzyme. Phospholipase C works on PIP2 in the plasma membrane, produce DAG and IP3 second messengers. IP3 binds to its receptor within the endoplasmic reticulum, release of calcium, response. DAG activates protein kinase C, protein phosphorylation, response. And yeah, that's pretty much it. Now, I'm not really sure why it says 3. It's definitely not a different mechanism. It's the same thing. It's just telling us how the calcium is going to do its effect on a new protein that is called calmodulin. So first of all, the calcium has to get inside the cell. One way or another, it has to get inside the cell, whether it's just by, influx from the outside, or it's by the method that we just explained, by the release from the endoplasmic reticulum through the IP3. But one way or another, I want the calcium to be found inside the cell. Now, as soon as this calcium is going to be inside the cell, it's going to bind to this protein called calmodulin. Now, the calmodulin is going to change its shape upon the binding of the calcium. Now, as soon as it changes its shape, it's going to activate other proteins. So, calcium calmodulin system, intracellular calcium, either from storage organelle, the endoplasmic reticulum, via IP3, or from calcium influx from the outside. Calcium bind to a protein called calmodulin. Calmodulin changes its shape upon binding to calcium, has 4 binding sites to calcium, and 3 is enough for activation. Now, by changing its shape, activate other proteins inside the cell. All right, and now for the final part, the lipophilic hormones. So now for the lipophilic hormones, as we said before, they get a VIP treatment. They don't need a transmembrane receptor. They don't need a second messenger. They can do everything by themselves. They're lipophilic. They love the lipids. So they get directly into the transmembrane, and then they attach to their receptor that is found in the cytosol. And then it's going to form some sort of a complex. And this complex is going to go all the way inside the nucleus, to a region called promoter region. That's a region that we've studied before. And it's going to induce the transcription of the DNA into mRNA. And then this mRNA is going to go out of the nucleus and into the cytosol and into a ribosome and be translated into protein. And this protein is the action that we're looking for. And, obviously this steroid or hormone receptor complex could induce this transcription process, and it can also inhibit it. Inhibiting it or activating it are both considered as responses. And obviously, the main examples of lipophilic hormones are steroids and thyroid hormone. So I'm just going to read everything through. Lipophilic hormones, as steroids and thyroid hormones, work by affecting the genetic machinery of the cell, transcription and translation, binding to their receptors in the cytosol, forming hormone receptor complex that will travel to the nucleus. Inside the nucleus, the complex binds to the HRE, the hormone receptor element, which is a promoter segment on the DNA. Stimulate transcription. Some hormones inhibit transcription. Produce single-stranded mRNA. mRNA leaves the nucleus going to the cytosol, and with the help of ribosomes, translation produce proteins, and hence the response. And that's pretty much it. All right, so this brings us to the end of the video. First of all, I want to give a special thanks to Drs. Abdullah Yash and Laith Nawaf. You guys are the best. Your summaries are incredible. I've been studying with them for the past 3 systems, and honestly, I don't regret it at all. And they have inspired me to do this video. I've always, explained in school, but I've never done it in university. But, and I know that I'm not the best at explaining, but I really hope that you've understood something, or at least just, enjoyed the video. Who knows? You might see me next time. I'm not really sure if I can do it again or if I will do it again. I just hope you're studying well, and good luck with your exams. And yeah, that's all.